So we are going to study today replication of DNA. For studying replication of DNA, first of all, we need to know the machinery which is involved in replication of DNA. Fine. So first of all, we'll study that how the various enzymes are required and how the enzymes function in the process of replication of DNA. First of all, we'll talk about the enzymes. The enzymes used are ligase, which helps in joining the two fragments of DNA. Fine. Joining the fragments of DNA. Fragments of DNA. Then enzyme helicase, which helps in unwinding of two strands of DNA. The strands of DNA are wound with each other in this way. Fine. So unwinding of DNA like this, it is accomplished with the help of enzyme helicase. So it helps in unwinding of DNA strand. Unwinding of DNA strands. Whenever we take any rope and we try to pull the threads like this, what happens? The supercoiling of another end of rope take place. So if supercoiling will take place, then process will not be able to accomplish smoothly. That's why a enzyme which is known as topoisomerase, is required to release the tension, or we can say is required to release the supercoiling of DNA strand on another end. So it releases the tension of DNA strand, or we can say that it releases the supercoiling of DNA strand. Now the most important enzyme is DNA dependent DNA polymerase. Fine. DNA dependent DNA polymerase. It helps in the polymerization of nucleotides while adding nucleotides in DNA strand. Okay, it helps in polymerization of nucleotides. It helps in polymerization of nucleotides. Okay. Now this enzyme have two limitations which are very important to study. First of all, this enzyme only function in 5' to 3' direction. It only functions in 5' to 3' direction. First limitation. Second limitation is that it is not it is not capable of initiating replication, or it is not, we can say that it is not able to gather the first nucleotide which is required in replication. Fine. So initiation of replication is not done by DNA polymerase. Now another enzyme, that is primase. Now what is the function of primase? Of course, when DNA polymerase is not able to initiate the nucleotides formation, who will initiate it? It is initiated by primase. But the nucleotide of RNA, which are known as primer, they initiate the replication of DNA. Okay, means primase enzyme helps in addition of primer. Primase enzyme help in addition of primer. These are the short nucleotides, or you can say the fragments of RNA. Of RNA. Fine. So these are all enzymes which are required in the process of replication. Now we come on to the machinery of replication, that in DNA strand which are the structures which helps in replication. Then first of all we'll talk about the ori site. Replication is not initiated from any site of DNA. It requires a particular site which is known as origin of replication. Origin of replication, or in short you can say ori site. Okay. Now these ori sites are present in vectors also which helps in the formation of recombinant DNA. Okay, so ori site is required. Secondly, the replication of DNA take place in certain steps. First step is activation of nucleotides, or we can say deoxyribonucleotides. Fine. Four such nucleotides are there: deoxyribose adenosine monophosphate, deoxyribose nucleotide, guanosine monophosphate, similarly thymine monophosphate, and then cytosine monophosphate. With the help of phosphate and enzyme phosphorylase in presence of energy, these monophosphates are converted into triphosphate. Okay, so adenosine triphosphate, guanosine triphosphate, thymine triphosphate, and cytosine triphosphate. Okay, now what is the importance of these phosphate? These phosphate basically they help in polymerization of nucleotide also. They provide energy and they act as substrate also. Means, see, they act as substrate. They also provide energy for polymerization. Okay, they also provide energy for polymerization. So this is the first basic step of replication of DNA, and after that we will study how the various enzymes which I have just told work during the replication of DNA. So first of all, the enzyme helicase. What enzyme helicase do? Enzyme helicase unwind the strand of DNA like this. Suppose this is 3' to 5' and this is 5' to 3' strand. They both are anti-parallel. And now this structure of DNA is known as replication fork. Replication fork. Fine. The unwinding is done with the help of helicase and prevention of supercoiling. See, this is unwinding and this is the non-supercoiled end of DNA. So here topoisomerase functions for preventing the supercoiling of another strand of DNA. Now the function of primase enzyme comes, and the first of all, so this strand is known as template strand. This is known as template strand. Okay. Or we can say them parent strand also. Fine. Either we can say parent strand or we can say template strand. A small primer is added with the help of primase, and this primer is removed afterwards enzymatically. Fine. And the strand, the daughter strand which will be formed, now it will be formed into the opposite direction of parent strand. If this strand is 3' to 5', then the newly synthesized strand will be 5' to 3'. Now this is primer. Initiation has been taken place. Now initiation is taking place. Okay. Now what will happen? Now polymerization of nucleotides has to be done. So how it will be done? With the help of enzyme DNA polymerase. It will add up nucleotides in the same direction by 5' to 3' direction. This strand is known as leading strand. It is also known as continuous strand. Why this strand is known as leading strand, that we will discuss right now only. Now this strand was in the direction 3' to 5'. Newly synthesized strand was in 5' to 3' direction. Okay, all okay with this strand. But now look here. The strand is in 5' to 3' direction, and the newly synthesized strand has to be synthesized in the opposite direction, means 3' to 5', and DNA dependent polymerase cannot function in that direction. Fine, it can only function in 5' to 3' direction, 5' to 3' direction. So now what will happen? Primer will get add up here. Fine. In which direction? In 5' to 3' direction. Okay. Now what will happen? Again, DNA dependent polymerase will come and it will add up here. Now again primer will come and stick here, and DNA dependent polymerase will start synthesizing it. Similarly, this whole strand will be formed in small fragments. Okay, this strand will be formed in small fragments. These fragments are known as Okazaki fragments. These fragments are known as Okazaki fragments. Okay, now this strand, because it is not continuous, it is in form of fragments. This strand is also known as lagging strand. This strand is also known as lagging strand. Now what will happen? Now these fragments, they will be sticked with the help of enzyme DNA ligase, or simply we can say ligase. Like this, ligase will come and it will stick the enzymes. It will stick the fragments. DNA ligase. Now both the strands, they will get separated, and in one DNA strand, one old strand will be there and one newly synthesized strand will be there. That means DNA is semi-conservative in nature. Fine. Try to understand what I am saying. Suppose this was the DNA strand. Okay, now I named it A and I named it B. After replication, what will happen? After replication, two strands will be formed. Fine. Two DNA strands will be formed. If this was A, then it will be newly synthesized A1. If this was B, then it will be newly synthesized B dash or B1. Fine. So they are semi-conservative in nature. They are semi-conservative in nature. Now the site of replication in cell, where replication take place, we should be known about that where is the site of replication. So replication, basically DNA is present in nucleus, and during cell division, one phase, that is S phase or synthesizing phase, is present, in which replication of DNA take place. During this stage, nuclear membrane is present, so the division take place, so the replication take place in nucleus only. Site of replication is nucleus. Site of replication, time of replication, when DNA is replicated, that I have also told. Synthesizing phase, semi-conservative nature, and this was all about replication. Thank you.